dynamics. Randy said my background is in theoretical physics. I've worked quite a lot in nonlinear dynamics, and we have spent the last 25, 30 years actually carrying concepts into the neurosciences and taking actually advantage of dynamics. Randy also mentioned EBRAINS and virtual brain. Virtual brain is one of our efforts to fuse concepts together and operationalize them in the digital neuroscience domain. This was about 15 years ago. And TVB, the virtual brain simulator, full brain network simulator, is one of the 3 large simulators in EBRAINS, which is European digital neuroscience infrastructure that has been created a few years ago, is permanent and accessible to all of you. So all the workflows that I will speak about can be accessed in eBRAINS and actually executed. Data are available, etc. Yeah? So that is useful. Yeah. However, just the tools and data available, that does not make yet any understanding. And you have seen causality. I mentioned the term causality in the title of my talk. Yeah? When you want to identify mechanisms that are underlying brain dynamics, you have to formulate a causal hypothesis. Causal hypotheses we phrase typically or very often in terms of a formal way. You can do this via mathematical modeling that you then have to interrogate. And this is actually becoming the modern form of doing data analysis, not just extracting structure that is in the data, statistical inference, AI, machine learning, but actually linking the data to a model, your causal hypothesis, yeah? And then extracting also information that is not in the data, that goes beyond the data, because your model represents your neuroscience knowledge. You link it with the information that is in the data and go beyond what is in the data, being able to make predictions that is linking or trying to make a best balance out of knowledge and data. And we call this inference, causal inference, in fact. But so, and this is a perspective that I will use. I will talk first about dynamics and some perspectives that we need to take seriously in order to be able to actually interrogate the brain dynamics that we are interested in understanding. And I actually going to jump in right away and show you here a simulation. And what you see here on the right-hand side is Kelly. Yeah, so next to Kelly, you see 2 time series plotted, yeah, that are evolving U and V. Yeah, it doesn't matter. These are just simulations. But whenever you have time series, you can actually span them in a space. You refer to this as state space. And in this state space, they are tracing out some trajectories. We should actually repeat. It doesn't. And what you see is that as the dynamics evolves, the trajectories go down to a particular subset. In this case, it's a circle. And there are subsections on this circle. We refer to this as a manifold. It doesn't have to be a circle all the time. But here you see that all the trajectories go towards the red points and away from the black points. What you're seeing here is essentially dynamics and that evolves over time. And the rules that govern these dynamics are represented geometrically in this particular state space. So you see a clustering of dynamics here on what we call a manifold. Yeah? And there is lawful behavior, namely once you're on the manifold, most of the time you spend on the manifold, you go very fast towards the manifold, but then slowly you follow the flow on the manifold. The flow is the realization of the laws that govern the type of dynamics. And that's your mechanistic formulation. Yeah? This is just translated into mathematics, and mathematics translated into geometry. At the end of the day, all of your data have a structure like this. It's not just a clustering of data, there is a directionality. And whenever you perform an analysis, you're actually analyzing the geometry of this particular object. We refer to this as structured flow on a manifold. Yeah? So every data set can be represented in this type of form. What you would like to extract is this particular information about the geometry of the flow, whether you know it or not, actually. Yeah? And then you can perform some analysis. You apply a metric like covariance matrix. What does a covariance matrix do to this? This object that we have represented here. Covariance matrix is an ellipsoid or second order statistics. So you're actually trying to fit an ellipsoid to the structure here, which means the amount of information, of knowledge that you gain out of this is zero. It's absolutely meaningless because you don't get this information here that you're interested in. Organization of the manifold and the flow. Have this in mind. I'm not saying covariance is useless. I'm saying know what you're doing in order to understand the response that the metric is giving you. Yeah? Could stop my talk here because that is actually the key message. Now let's use these concepts. And the rest of the time, I wish to share actually with you how we are working with that. Yeah. So first of all, why is that meaningful? Structures flows on manifolds are a representation. Oh, you're not seeing this part here. Yeah, would you mind? How do I get rid part? This one here? Let's get rid of this because you're seeing different running patterns here. Yeah. A structured flow on a manifold, what that is, it captures details of primitives. In running, it's motor primitives. It's just clumsy in running. Yeah. Sorry, the repetitions are not working. But bottom line is, when I spoke about the lawfulness that the structured flow, the geometry of the flow represents on this, this is at the end of the day, the type of representation of understanding of what behavior, in the most general sense, what it actually means. Yeah, behavior. Here I'm citing a good friend of mine, Scott Kelso, is not just a particular time series. It's a particular realization. What it is actually it's a set of all possible time series, as you have seen here, which samples the manifold, that is actually extracting the coordination laws that are underlying this particular behavior. Can be cognition, but in motricity, you see the easier represented. So what we want to know, if we're making the link between behavior and brain dynamics, we need a good representation of the dynamics. We are proposing is this is the structural flows of manifolds. It's actually a no-brainer. The problem is how do you deal with that? Yeah? And when you go in the literature back last 50, 60 years, there are different forms of self-organization because it's a theory of self-organization that is actually underlying this emergence of the structural flow on manifolds. What it is it's meaningful, lawful behavior, which means it must be low dimensional. Yeah? Technically speaking, it means you're in a high state space and you have the emergence of these manifolds, which describe certain patterns. Here, what you see is actually a famous example from self-organization. That's a Rowley-Benard experiment where you have a fluid heating from below and you have pattern formation arising because particles are being moved up and then in roll patterns go back down. This is one of these standard examples in theory of self-organization. And you see this. So you, in a very large high-dimensional system, these type of low-dimensional patterns emerge. This has been in the 50s, 60s. There are people such as Ilya Prigogine, who received the Nobel Prize for the explanation of this type of pattern formation, or my former mentor, Hermann Haken in Stuttgart, yeah, who's a pioneer in and the creator of the theory of synergetics. Mathematically formalized it in the sense that whenever you have a complex systems, high dimensional, with nonlinear interactions, you have the capacity when the system is being driven from the outside. So you need to open the system. Otherwise, it follows the laws of thermostatistics. Yeah? If you drive it from the outside and the interactions are nonlinear, then there are certain conditions under which the formation of such patterns occurs, which corresponds exactly to what I showed you earlier. You have a collapse of the dynamics on a low-dimensional manifold that you see characterized here. And he called it order parameters that are then enslaving, which is essentially what it means. It predicts the behavior of all the other degrees of freedom, because you must have an information compression somewhere. Yeah? So the theory was extremely powerful, predicted the laser activity, explains superconductivity, explains how ice is being formed, the transition from water to ice, etc. Many patterns in human perception are being described by that. The shortcoming here has been that it's valid only in situations where you're actually close to the onset of the transition from one behavioral state or from one state to the other. Yeah? That is the shortcoming. And there has been work in different domains, and here in this case, in brain connectivity also, that can actually generalize this and say, actually, there are also other conditions under which you can actually have this compression of information. This happens here. Imagine you have a connected system where you have an all-to-all connectivity. Always, when you have an all-to-all connectivity, you're creating, in every case, this type of circular, spherical manifolds in state space. And then you have a small form of symmetry breaking, as you see represented here. Here I'm showing you a manifold. Everything is contracting towards it, but there is no flow. And through the symmetry breaking, you're actually generating flows on the manifold. Remember, the flows predict the behavior of the system. These are the rules that are underlying the dynamics of the system. This is what you need to know. This is actually what you want to extract, because these are the mechanistic elements that you would like to understand. This happens in the neighborhood of symmetry breaking. And there are many symmetries in the system. Here, connectivity is just one of them. But it's important that you do not need to tune the parameters for a system to have this reduction. Yeah? So it's actually omnipresent, in particular in biology. So what I want to do is I want to show it to you one more time, the geometry. Whenever you have a system, and in this case, it's just 2 degrees of freedom, X and Y, 2 degrees of freedom, they are following an arbitrary dynamics. When you have all-to-all connectivity, you always generate this type of dynamics. And then you have a symmetry breaking. And this means you are destabilizing, or not destabilizing, you're introducing a perturbation that actually generates these one fixed point, stable here, unstable. This is just a toy case example. And that generates then this flow along the manifold. Always, you can actually demonstrate this mathematically. Or you can have multiple points. Yeah? But this is exactly the type of dynamics that we were talking about. And this is what the symmetry breaking does. And if it's small enough, then you have a channel in here in which this dynamics can evolve. So this is the simulation I showed you earlier, yeah, with the multiple fixed points on there. So maybe you can appreciate it one more time. Now let's drive it with noise, just one single trajectory. What you see is actually in a biological system, you have noise, it's omnipresent. It samples actually the geometry in the form of the manifold. You have deterministic features in there, which is a flow. And the noise is sampling the geometry of the manifold. Whether you want or not, whether you're accepting this as a framework for thinking, yeah, the features, you have them in your data. The deterministic features that are representing these causal aspects, and the noisy features that are causing this sampling of the geometry in the state space. This was only 2-dimensional. You can do the same thing in 3 dimensions. Actually, this here's a simulation, 20 dimensions, and the manifold was created to be 3-dimensional. On the left, you see that you have 2 particular fixed points, tack, tack. Then the noise kicks it out and goes over there. But I'm showing you here by putting the sphere in there, you can actually appreciate the geometry of this. Yeah? Just looking at this, you don't know what's happening, but putting it into the context of these type of geometric objects, it's actually quite obvious. Yeah, two fixed points, one separatrix, so like a wall that separates these two, all compressed on a manifold, 20-dimensional state space. Yeah? So the question is, how do we work with that? Yeah, how do we deal with that? Theory on the one hand side, and on the other hand, are there examples in the literature where people are doing this? There are many, in fact, manifold. When you look at that, at some point in the 90s, it was chaos. Yeah, then became complexity. Then it became connectivity. Yeah, manifold is one of these other keywords that you find very, very often in the titles of a large number of publications that is upcoming now. This here is a lovely example from C. elegans experimental data. One of the challenges is actually how to extract these manifolds, yeah, and how to sample them. And this is just an example of experimental data. They're using some techniques that are linked to information compression and a nonlinear PCA, so that they are sampling the manifold and they are linking it then actually to observed neuronal activity they actually measured in the organism, and then is reflecting the different type of behavioral patterns, forward locomotion, backward locomotion, so oscillatory pattern, etc. And that corresponds to sampling of individual pieces, individual elements on the manifold. This is coming from experimental data and then can be represented in the simulations. And with this type of thinking, yeah, I would like to invite you, okay, can we do the same thing now for taking the same approach, thinking underlying lawful behavior, so structured flow on manifolds, when we analyze the data, and can we design experiments in order to make sense out of some data that we have measured that are relevant for certain applications, either in function or in dysfunction, so clinical translation. And that's what I wish to walk you through. First, a few words on the virtual brain, because it's our framework that allows us to formulate a hypothesis. Yeah. So the virtual brain is a computational modeling framework, and it goes back by now about 15 years. That was a grant that Randy was leading at the time with the brain network recovery group. And we have designed an approach in which we actually fuse empirical data with mathematical representation. Yeah? You take an individual, you image the individual, you extract structural data, because we want to have a constraint imposed upon what we will formulate as causal mechanisms. So it starts with connectivity. Remember, I spoke about connectivity before. DTI provides us the connectome, the large-scale connectivity matrix, geometry, so topographic information we get from the MRI. We piece it together in the same brain reference space, and then we performing a parcellation allows us to span a network in 3 dimensions, in 3-dimensional physical space. Each network node, and here with about 200 network nodes, we have about 10 to 15 square centimeters of surface space covered. So that's actually pretty big, but this is the level on which I will speak about. What we can do today, we can go up to 250,000 parcels. That provides us with a resolution of about 1 square millimeter. Yeah? But this is high resolution TVV, which is not the topic of today. Yeah? So here we have 164 actually network nodes, where each network node we equip by a neural population model. This mean field theory, this is concepts of statistical physics that are being applied to Hodgkin-Huxley equations, you know. So individual equations of single neurons, you build a population, and you perform statistical averages that are meaningful for the application that you wish to obtain. Then you get actually reduced equations that are describing collective variables such as mean firing rate. Yeah? Assuming that mean firing rate is a meaningful realization. And it is in most applications excitatory, inhibitory, and then we get reduced mean field equations, typically 2, 3, 4 degrees of freedom. That's what is being referred to as a neural mass. So you equip essentially each network node by such a neural mass, you use the connectivity to span the network in 3 dimensions. And you can write down the mathematical equation to run independent network simulations. Even more importantly, to bridge the gap between source activity. No one in the world can measure source activity. You always measure sensor signals. Yeah. So we need to make the link between sources and sensors, and that's how we do it by adding the so-called forward equations to it. So once again, in 3D, we parcellate, we build a network, we connect the individual network nodes. These are then our source activations, and then we put in our sensors. In EEG, for instance, stereotactic EEG, we put in invasively electrodes that in this case are 1, 2, 3, 10 electrodes. They go deep, allow us to measure deep sources. And typically we have 10 to 20 electrodes. So we have a sampling up to 200 points in there, or EEG, MEG, or fMRI. Yeah. So this gives us the additional link thereof. Yeah. This here is a snapshot of the platform that we use in the virtual brain. And the virtual brain is a simulator that is inside of eBRAINS, so you can actually access them and operate it inside of eBRAINS. The other simulators are Neuron, that's a blue brain simulator that you have heard of probably, and NEST, which is a reduced neuron model. So we are going all the way down from the hundreds of micrometer scale, millimeter, all the way to the full brain. And then through this, you can actually simulate the type of activations on the source and on the sensor level as I just described. Here, just in this example, EEG. So that's just for visualization to show you what you can actually do when you download the system. Yeah. So let's take this approach now. It's a virtual brain modeling approach where we use individual data. So there's already a degree of personalization in there. Please recognize that personalization, inter-individual variability is one of the largest obstacles that we have in clinical translation, yeah? Because you want to make statements about this particular patient, yeah? And not on the group average. Yeah. So I will walk you through 2 examples. Yeah. One is physiological resting state activity. So you could argue it's not very functional, but this is essentially what we do when we look at cohort studies. Yeah. And the second one is in epilepsy, pathological manifold. Keep the idea of manifold in the mind, because we have to look at that data. When we look at data, resting state is particularly useful, and here we look at functional MRI data. And Sam, can you explain to us later what fMRI is, so I don't have to go into this? Thank you. It makes perfect sense to give you a talk like this, and afterwards we explain to you all the things that I'm not explaining. Resting state. Functional connectivity, for instance, is one of the markers that is being used over and over again. Essentially, it's a covariance matrix of, remember the individual sources that we have? You compute the covariance of the functional evolution of your hemodynamics. This is what we have when we measure fMRI, then we quantify it by functional connectivity. It's meaningful, yeah, beautifully, and you find differences across groups, so it's being used as a marker. All the way, aging, biggest difference, age and sex. If your metrics cannot distinguish between female and male or over age, it's useless. Yeah. However, you find epilepsy, obesity, psychiatric diseases, you find on the group level loads of reasonable and justifiable differences. But when you go on the individual level, it's typically useless. Yeah. You cannot make individual predictions. Again, keep in mind, manifold data distributed in a space. What does functional connectivity do? You see there is a structure in there with the flow of the data. What you're fitting is an ellipsoid to it. So if the data organization is not such that you can fit meaningfully an ellipsoid to it, for instance, that you have 2 fixed points, one here and one there, and then you have a sampling, then it's absolutely meaningless. Yeah? Especially if your data cloud is kind of circular, spherical, you will get absolutely 0 information on the individual level. And that's one of the parts that we are suffering from. The other part is most likely it's highly non-stationary. And then I will show you examples for that. Yeah? And non-stationary in a finite time window, it means that you're sampling only individual subspaces. And there another large part of variability comes from. Just an example from aging, I will speak a little bit of aging, as there with using functional connectivity and structural connectivity, you have very meaningful predictors. Yeah, for instance, here from this study, yeah, plotted over age, when you look at the structural connectivity and how the streamlines are distributed, there is some predictive power. The same thing for functional connectivity. Yeah, but when you try to put them together, trying to figure out how structure constrains function. Remember causal hypothesis, yeah? So some mechanistic insights. On this level of description, you're learning absolutely nothing out of this because there is actually no meaningful correlation. So you need to have a more informed approach to extract this type of information with insights out of the data. So here I'm showing you two examples of subject 1 and subject 2. Their connectome, structural connectivity, left hemisphere, right hemisphere, interhemispheric connections, structural. Same here. Then you measure the hemodynamics and plot it over, in this case, it's about 10 minutes. And you see the fluctuations for some selected brain areas. What we typically then do, you compute the covariance over all areas, left hemisphere, right hemisphere, interhemispheric. You get your functional connectome. Subject 2, subject 1. When you do this on sliding windows or non-overlapping windows in this particular case, you see that there is a large variability here. This is actually something that we are suffering from. And this is one of the reasons why we're on the individual level, it actually does not work very well. But you can actually trick, especially for the young ones, when you run into an obstacle and you have a major problem, yeah, and it's destroying your analysis, your modeling approach, use it, operationalize it, weaponize it, yeah, and make it actually your tool in order to interrogate the system. Here, the non-stationarity is a problem. Let's use the non-stationary to make predictions. And one way of doing it is actually you can compute the functional connectivity per time window, per subsequent time window, subsequent, and then actually compute these individual functional connectivity matrices and correlate them. And then you get something like one FC correlated with the other FC. Cross correlation gets you 1 point here. So here the axis is seconds over seconds. And this is now a cross correlogram of functional connectivities. So on the whole brain level. We refer to this as FCD, functional connectivity dynamics. And what you find here is you have, look, this was 10 seconds or 20 seconds actually. I plotted only 10 seconds, sorry, 10 minutes. This is 20 minutes. So you have areas of minutes, 2-3 minutes long, where the functional connectivity, so the set of covariances does not change. And then it disintegrates and reorganizes again. So we have periods of stationary invariant functional connectivity, where the second order statistics does not change. You can actually now use this and characterize this type of fluidity, these changes in the brain dynamics of the subject, and apply it across age. And here, just to give you an example, young, medium age, older age, so senior. These are different scales where the time window of the functional connectivity, 60 seconds, 30 seconds, just to show you that actually it scales. But what you see already with the eye is that there is a loss of fluidity, or said differently, you have more time spent in the same subspace on the manifold. Note how I'm using the language, because same subspace of the manifold implies that the covariance in which the system is actually does not change. You're stuck, not stuck. You're occupying this subspace longer, whereas when you're younger, you're moving much more fluidly around. This is one way of putting this example, this together. When you do this on an individual level, on the subject age, you can actually become very predictive for the age, chronicle, Yeah, I'm stuck in the manifold. Yeah, thank you. The predicting the age on an individual, chronological age. Thank you. Yeah, and the interpretation is essentially the following: you are stuck on a manifold. That was you who said this, right? Going here and over there. Yeah. So whereas when you have no sequential correlations, it averages out here. Yeah? So you can quantify this, for instance, by jump length, the structure that you have in here. So we can actually operationalize the non-stationarity that hampered us before, now make use of this on an individual level. Yeah? In order to understand this, we can go even further. For each individual subject, we can build the virtual brain, yeah, and actually use the individual connectome. And then we need to modulate it. We need a working point for the strength of the connectivity. This is what is actually represented here, the global coupling strength. And we can plot now along this axis the global coupling strength, which is a measure for the degree of neuromodulation. Typically, we use here dopaminergic systems, subsystems, and the noise strength. Remember, in the simulation, we can introduce noise that is then also doing the sampling of the manifolds. Then for the different parameter places, we can plot this FCD. Two forms of FCD. Rennie, for you, cascades. I'm not talking about cascades. FCD for this, FCD, you see the structure emerges that we see with more fluidity, and then it starts disintegrating again. There is a preferred regime in which the brain fluidity is actually the way we have seen it in the healthy data. And with increased age, the brain fluidity actually reduces. Yeah? But this is coming from simulations where the simulation does not uses only the structural changes in this particular area. When I'm projecting the manifold, we can actually extract from the data in a two-dimensional space. We can estimate the dimensionality of the manifold. It's about 6 to 8 dimensional. Here, just two-dimensional projections. I'm showing you the corresponding manifolds, how they change their shape, and actually the density and the occupation time of individual areas. But again, 8, 9-dimensional space, now projected into 2D. So with the eye, without algorithmic methods to characterize this, we don't get much further. But we can do some simulations in order to give you an idea. Here, principal component analysis for the manifold. We are running the simulation. We're computing the FCD. Remember, FCD is time over time cross-correlation, where there should be something that is going through. Was the simulation ran? Yes. Yeah. Yeah. So I do it here. FCD, do you see? The cross goes through. And essentially you have, during certain areas of these squares in here, you have an occupation of this set of the manifold, and then it jumps over there. And here we're plotting the functional connectivity, in fact, averaged across a time window that's sliding through that. So out of this, you don't get anything, but you can actually show how you get subsections of the manifold occupied during these periods of invariant functional connectivity. So now how we can make use of this in order to understand better what is happening in this domain and linking it to causal hypothesis. We essentially have to change the hypothesized aging mechanism, the structural features, in order to constrain the mechanism that is behind there. Can analyze the structural data that we have. Yeah, it is known that there are structural changes, in particular in interhemispheric changes. That was when we go to the Jülich database. It's a 1,000 brains cohort in Jülich, a beautiful aging cohort from 50 to 80 years old. 750 subjects, despite the fact that it's called 1,000 brains. You can actually analyze the structural data. Interhemispheric connectivity goes beautifully down, all 700 subjects plotted here in this domain. And you can actually build a mask, left-right hemisphere, taking the callosal fibers, because there you have the biggest degree of degeneration. Yeah? So you can extract that out of the data and actually start taking the young subjects here around 55 and start virtually aging them. You can actually build the aging mask and start in the brain, in the virtual brain, degenerating the callosal fibers exactly the way we see it for the actually really aging subjects. This is what we do here, and then we can build cohorts and start seeing, can we actually capture the type of functional data using this mechanism in order to explain what we see? And the answer is, this is essentially what I just explained to you. We built the virtual cohort out of the individual subjects. We introduce a virtual aging mechanism, and then we predict the different types of metrics, functional connectivity and functional connectivity dynamics. Yeah. So, and these are the results that we get out of this actually. When we virtually age the young ones around 55, and then the virtual aging goes in this direction, we still need to, we have another parameter left, that is a neuromodulation, particular here, the dopaminergic subsystem, the neuromodulation that we can vary here. And we do a full scale parameter sweep. This is the type of behavior that we see. When we then take methods from machine learning and fit actually every single subject now for his or her own connectome, we get this type of behavior here, and actually mimicking the effects that we see for the virtually aged subjects. Careful, this is a subset only at 55 age, 55 to 57. We are seeing actually that there is a compensation mechanism that would be predicted from that, actually compensating in order to, by increasing the neuromodulatory effect in order to go into the direction of maximum brain fluidity. Yeah. So this is what we get out of this, and this, when we, so purely aging would go on the structural data, you would go vertically down. Yeah. But then you find that the maximal brain fluidity you find here. You find the same thing for the empirical data set here, for each individual data, not group level, but each individual data set represented here. Then you can correlate it actually with cognitive performance, and you find actually a clear statistical separation that those that compensate stronger towards the maximum of brain fluidity are the high performers in the cognitive testing that was performed for the entire cohort versus the other slow performers. Randy, how much time? Still 10 minutes, I think. Yeah, thank you. So having said this, with our capacity of virtually aging and introducing some other mechanistic aspects through this type of mask approach, where we can actually constrain hypothesized mechanisms, we can actually do the following. And this is what Randy and I are developing as a research program. We can estimate, this is after he was down on the hill and happy to be down. You can estimate the current state of health of your brain at a particular point. Yeah. And then because we have a predictive model, a causal model, you can now actually start estimating the brain health trajectory that you're linked to cognitive performance, yeah, that your brain will undergo in the next 10 to 20 years. This was a range that we had, yeah, depending on particular manipulations as reflected in brain, in the characterization of brain activity that I just showed you. It's his discriminative for certain brain diseases. Yeah, so we're working with schizophrenics at the moment. Yeah, a large cohort in Marseille. Yeah, where we have worked only here on this particular level, but along this axis, because you have a causal hypothesis, you can actually start making projections, starting where your initial time point is, in which direction it will evolve. Yeah, it's always a range of trajectories that the system can take. So this is an ongoing research program, yeah, that is in its beginnings. Where we're much further evolved is in epilepsy. And in epilepsy, we have worked on for the past 7-8 years, and it is actually our workhorse for the clinical translation, taking these concepts. The mask, the virtual aging mask that I was talking about, this was this concept of a mask that provides structural constraints based on the connectivity of an individual brain. In epilepsy, it is actually the epileptogenic zone where it's located. This is a mask that we wish to estimate. I'd like to show you how we are actually doing this. Yeah? So first of all, epilepsy. We are focusing on drug-resistant focal epilepsy. Look at the literature. 1% of the human population is epileptic. Of those, 60% of all the epileptic patients we can treat pharmaceutically, which means a third of the patients we cannot treat pharmaceutically. Typically, there is an evolution. Different medications are being tried. After a few years, the patient is being considered to be candidate for surgery because that is the only hope for relief of the particular patient. Yeah? In order to explore the patient pre-surgically, we use stereotactic EEG. Yeah? That's a technology that has been developed in the 60s by Jean Banco and Jean Talarac in Paris. Yeah? You saw the needles earlier, so about that long, spacing of 2-3 millimeters. You drill a hole, you enter the needle, and you start exploring. 10 to 20. When you're in Milan, 20. When you're in Marseille, 10 to 15. So there are different approaches. This is a type of dynamics that you observe. Yeah? Pre-ictal activity, onset of a seizure, paroxysmal, high frequency activity, not always large amplitude, as you see it in this particular element. The idea is to understand, the hypothesis is there is an epileptogenic zone, which is at the source, at the origin of the paroxysmal oscillatory activity that then starts propagating and recruiting other areas. Therapy is you enter into the brain and you remove the epileptogenic zone if it's removable. So you always have to find a balance between cognitive deficits that this may cause, safety for the patients, and benefit for the patient. Yeah? But diagnostic of EZ, epileptogenic zone, and therapy coincides in this particular case. Data feature reasonably well to be quantified by a metric is this one, state space spanning a manifold. The question is, what is a pathological manifold that you actually want to sample and estimate that should then be informative for this? It reduces to the estimation of the epileptogenic zone. Very important, because when you look at this, over the last, it goes actually back to the 80s, over the last 50 years, surgery success rate, if you average across the different types of epilepsies, it's around 60 to 70%. Yeah? Which means 30 to 40% of all the failures. Yeah? So essentially, this is work that has been done now across different species. This is done with my colleague Christophe Bernard in my lab. In mouse, hippocampus in tutu. You extract the hippocampus, yeah, and you render it epileptogenic, typically with low magnesium conditions, which you see represented here. What is being mapped is local field potential. This is multiple minutes, in fact. Fast discharges, interictal activities, fast discharges, pause, fast discharges. It's being accompanied by extracellular measurements of potassium. So there is a slow dynamics. You will see in a second why that's important. That accompanies that. It's a multifactorial disease that is actually tracing out the dynamics of the seizure. In the 80s, 90s, by Uwe Heinemann at the Charité in Berlin, he has hypothesized that this was one of the major contributing mechanisms on the extracellular level. Also energy, so ATP that was measured, oxygenation. These elements are all connected, but we can pin it down better. It seems to be best represented by extracellular potassium. We can zoom in here and you see the details of the seizure being traced out on this. Back to the manifolds. When you have a so-called fast, slow system, you can always represent the state space by its fast variables and the corresponding slow variables. When you do this for something that looks like this, it will give you a spiral. This is the interictal activity. And then you have seizure onset, fast activity, and back down. Yeah? It's characterized, so it essentially represents this activity. This is a time series. This is a corresponding manifold. And in order to have a systematic characterization, we talk about bifurcations, is essentially the rest state or interictal state needs to destabilize to jump on this particular level, fast discharge, and then it gets exhausted, but mathematically speaking, you undergo another instability back here. We were able to demonstrate, and it was verified by many labs and sampled in many different species under many different conditions, that this is the geometry, pathological attractor in epilepsy. Yeah? It's not that simple. There are a few other things. However, now we can actually take this pathological manifold, formalize it mathematically, that becomes our network node system, and go through the type of virtual brain dynamic workflow that I described to you earlier. You construct the patient's brain, span it, virtualize it, equip it with the corresponding mathematics. In this case, the pathological manifold that has rest and pathological discharge state. You can refine it by additional information you get from structural MRI. And then you can use it for fitting. You can use it for simulation, but you can use it as a fitting target. Remember, causal inference. You put model together with data, then you are able to predict not only what is in the data, but also what goes beyond the data. Yeah? And predictions going back to the epileptologist. So this is what we did. Here just in simulation of a patient. You can perform individual specific implantations. And you can estimate the epileptogenic zone. And you can actually run individual simulations and start performing different types of treatments. What if I do not measure here? Yeah, can I see something? What if I perform virtual surgery? Yeah, which is actually what you do in the clinic. Yeah, you can use it as a platform in order to do this. And this is what is now being done systematically in different reference centers in France and Italy and in Pittsburgh. So these are our collaborating centers. We need to estimate, this may be a technicality, the epileptogenic zone here overlaid with the post-surgical MRI of a particular patient. This is what we want to have. And in addition to this, it's not an optimization approach. We are sampling the manifold and also its parameters, which means for each brain area, we can get a sampling of the possible and most likely parameters corresponding to its epileptogenicity. So we're not just estimating the maximum, but we are sampling the entire range of all possibilities. This is very important because this is experimental data. Sometimes, or very often, it's actually bimodal, and there are multiple possible explanatory hypotheses for the same effect. Here you see an example where a patient in red, epileptogenic zone overlaid with a post-surgical MRI. This corresponded actually well with the surgery that was finally performed. And here you have an example where it did not correspond. So there was a large discrepancy between the surgery that was performed and what came out of the virtual brain. So we can actually, this is how we are actually estimating this. Retrospectively, we go to patient cohorts and we measure the degree of discrepancy between what is being done with the virtual brain and what was actually executed. This is done for a cohort of 54 patients. So essentially, the performance is not bad. Yes, you know, 60% for precision, which is equivalent to specificity. Important actually, and other metrics, AUC, you may know, 76%. When you split it into seizure-free patients, 1 year seizure-free after surgery, yeah, and look at the performance, this is what is from the clinic, coming from the clinician, clinical hypothesis. This is coming what virtual epileptic patient from TBB. It's actually equally well performing. However, for seizure non-free patients, it's actually dropping down a lot, in particular for the TBB, which tells you when there is a large discrepancy between what has been done, really executed for the patient in terms of surgery and what came out of TBB, often you have a larger tendency towards non-seizure freedom. And we actually looked in more detail into this for the retrospective activity. And we find when we again look at seizure-free, not seizure-free, the performance drops down. So that was reinsuring again for a large patient cohort, 54 surgery patients, that this is actually a reality. And then we looked into what is actually happening. With a virtual brain, we can actually simulate every single brain area. But the clinicians, when they perform their analysis, they have only access to the areas that were actually sampled by the SEEG. The subjects that had the biggest percentage of surgery failure were typically those where from the virtual brain, it was predicted to be part of the epileptogenic zone, but not part of the sampling. So the clinicians actually did not have access to this. Yeah? This is now being tested in a large clinical trial, systematically with 400 patients that is ongoing in France, yeah? Covering the 13 reference centers in France, where they are undergoing actually the virtualization and the prediction where they're using it in the decision-making for the surgery. Today, this started about 3 years ago. COVID slowed it down, but today we are at around almost, most of the centers were closed for a large period. Yeah? We are at almost 300 patients, more than 50 surgeries have been performed. We will reach the 400 by summer next year, and then we will essentially be able to touch these data and perform. Yeah, I'm done actually, Randy. Yeah. Okay, 5 minutes. No, seriously. So we will be able to close patient inclusion summer next year, and then be able to work with the data. Before, we're not allowed to touch them for obvious reasons, because we cannot interfere with the clinical trial. So that's what I wanted to share with you. Essentially, I wanted to share with you, I was asked to speak about brain dynamics and share some theoretical concepts, theory and concepts with you. That's what I did. Structured flow and manifold is actually the language of dynamics. And we need to go beyond statistics. So dynamics is not just a data point distribution. There is a directionality, a sequence of that. You saw how important that is. Yeah? From the theoretical domain, when we exploit that, in aging, we used FCD. And in seizures, you saw the evolution, the propagation of the activity. That's a highly non-stationary phenomenon. You must have a representation of the dynamics. This you can do geometrically by plotting or understanding what the manifolds look like. Admit, it's extremely difficult when you have a data set. How do you extract the manifold? It's a very active field in statistics and machine learning, essentially, to extract and learn these manifolds that are actually determinant for the dynamics. Yeah? What is being done here, and that's also a form of digital twin technology that is actually what was developed in the 90s, 20s by NASA in particular, yeah, where the simulation platforms come from. Yeah, they are feeding in data, real-world data, into their simulations in order to adapt their routine, and then feed back the data into real-world applications. So this is what we are essentially after. We are fusing data, structural data in particular, to constrain the mechanisms we have in the model representation, and then run simulations, if possible, in real-world interactions. Yeah, stimulation, brain stimulation is one of the active fields that we have. Some colleagues of mine that work in robotics and spatial navigation use this to optimize some of the procedures. In our hands, for instance, to optimize stimulation protocols for individual patients, and then feed it back into clinical real-world applications. This is what we do. This is a vision also for the future, because you can also map now 10, 20 years in the future and see how far can we go actually with virtual and digital representation of external environments and with the details of the mechanistic models that we do. But key is actually, we are not able to build a twin of the brain. We can specify it for certain specific questions that you're asking, as we have done it in epilepsy, for instance, in order to get a particular answer on the individual level. Yeah? So don't think copy of the brain. No, no. Digital twin technology does that for a specific question. This is what we're actually pushing. So I thank you for your attention. I thank my colleagues and friends that have accompanied me for many decades, actually. Some of them, Randy and I, go 20 years back. And I thank our funders. Thank you for your attention. I stay here? Yeah. I stay there.
Well behaved. Thank you, Victor. We have time for questions before we break for coffee. Todd, please.
That was a wonderful talk, really inspirational. I have so many questions, so I'll just start with I have two that I'd love to ask right now. The first one is that graph that you showed where there's PCA1 and PCA2, and then there were points all in the scatter plot, and then you over time sort of highlighted each one of those points.
It was for the aging, the resting state manifold. Yes. What is it?
Well, the question is, do you have an interpretation for those PCAs? I couldn't really understand what they were.
Yeah, I can say this. In this case, it was just a naive principal component analysis. You need to visualize the manifold in a good way. This was an old movie. I have this old. The way we do it today is we want to span the subsystem in which the manifold lives. How do you find the manifold? How do you span it? PCA is naive, but it's very fast. Yeah. So that was that. Today we use more elaborate nonlinear procedures to find where is the maximum of the density of the points, because this must be where the manifold is, and then be able to span this. There is a nonlinear PCA. There is a procedure that is called TICS that is estimating actually the manifold itself. But the PCs, we just want to span the space in which the manifold is. You're not spanning the manifold itself.
So the interpretation of the PCAs, not interested in. What you're interested in is the interpretation of the movement around that plot, and that's the manifold.
Yeah, the manifold is essentially the structure you have there, geometry, we called it earlier. And then the dynamics I have to show there because there is a directionality flow on the manifold. It's not just a data cluster.
There's not a way to quantify that?
It is, but we're struggling. That's why I said manifold techniques at the moment is a hot topic in this domain. Whenever you want to span a manifold, you have to make certain assumptions because there is noise in there. You reduce the noise to get rid of the noise. Like this, you're already tinkering with the data. So whenever you make a reduction for visualization, you're making an assumption. Yeah. So this needs to be justified. There was a paper just 2-3 years ago, very recently, a technique that is trying to optimize these different techniques for the manifold extraction. I can send it to you. I forgot the name of it. It's PACE. It was in Nature Methods. So there are techniques. It's non-trivial, and all of them have certain assumptions to do exactly what you just proposed.
And the last follow-up of that, if it's 2 dimensions or 5 or 10, it doesn't matter to you. It doesn't get more complex to whatever to extract the manifold. You had 2 PCAs there.
But I visualized only 2. Yeah. It was what did I say? 6 to 8 dimensional system. I'm incapable of visualizing a 6 to 8 dimensional system.
Mathematically, just
cross sections, yeah, or you can use color. The beyond 3D, 4D with color, time, and maybe a little bit of motion. You can visualize a 4-dimensional manifold, but these are the limits. Yeah.
Any sense of amount of data required? Have you explored or have a sense of how much is needed for digital twins? I gather that to constrain your models, you need a certain amount of good data to display that digital twin. Any sense of?
There is no good general answer. We had a workshop in Paris a few months ago on digital twin, perspectives, and etc. People were fighting half a day. Jean-Pierre Changer was saying, digital twin, rubbish. Yeah, you don't know the mechanisms, you don't know how to do that, you will never be able to have a digital twin, it's sci-fi. Then Albright came and said, rubbish. Look, your digital profile on the internet is a digital twin, yeah, it's useful, yeah, etc. So worlds with 2 extremes. Digital twin exists already, others say digital twins will never exist. The point I take is it needs to be specific for the question you ask, and it may make it more useful. The way we are making it useful here in epilepsy in terms of we are able to constrain it with about 200 dimensions. Yeah? So this is sufficient. You're asking for data now, how much data we need for an individual subject. If we are asking questions about organization, connectivity dynamics, about 200 nodes are sufficient. You should have structural connectome, and then you have a good enough predictive power that goes beyond generic brains. This has been demonstrated over and over again. If you're interested in aspects outside of epilepsy, so with more difficult dynamics, metrics to work with, I would recommend sometimes higher dimensionality. Yeah, but it depends on what question you want to ask. Whether it's abnormalities, deviation from the resting state, I would actually not go from the major mainstream, what people consider to be healthy rest. Yeah? In psychiatric conditions, for instance, I would not go too high dimensional because the curse of the dimensionality comes in. So I cannot give you a general answer how far away we are. The digital twin, I personally believe it's a good approach, a good technology that we can exploit, but we need to render it specific for the type of application. I know you're interested in brain stimulation. Brain stimulation, the region of interest should be at high resolution because you need to know about the coupling between electric field and the corresponding tissue. But the rest does not have to be high dimension. This would be a potential design for a digital twin in applications that you may be interested in.
One more question, last question, Sylvain, please, on Zoom.
Hi, Victor. Sylvain Morneau here from SFU. Thank you very much for this very impressive talk. Thank you. Victor, I graduated from Marseille, so it's great to see you here. That's awesome. As you can tell with my accent, probably. I work mostly on aging and trying to understand how we can understand the factor of longevity in aging. And so that's a little bit linked to my question. I think the work you have done with Randy on aging is very impressive. And I was wondering, and this is probably like a very general question, but where do you see the limit or the next challenge in the virtual brain in modeling aging?
The limit or the next challenge?
Yeah, let's say both.
The way where it's a general question, and for sake of time, let me give you two answers in terms of challenges. Number one, it's a little linked to the last question I answered. I would like to see high resolution because interventions with the virtual brain are of major importance. I'll give you an example. Resting state activity has its limits of being informative. Yeah? What you're interested in is function, maintaining or reinstalling cognitive function or function. So you would like to have functional tasks. They are often not that well controlled. So what you can do is, if you want to go beyond resting state, is you look at stimulation paradigms, not for therapeutic purposes, but for diagnostic purposes, in order to sample the dynamics that a particular individual has in his or her brain. Yeah? So for this, you need to go with the virtual brain into high resolution and start getting a better understanding of stimulation. That's one aspect. The other aspect is we have a huge amount of data available that are being collated and put in the same reference spaces. If they can be mapped to be interoperable, yeah? And for instance, having the neurotransmitter distributions across individual brain regions, there is a large variance of which we know it is informative. So it will actually affect the predictive power. So there, I would like to see that when we have high resolution representations, that we can actually go to an implementation of more detail, not necessarily on the microscopic level, but from one brain region to the other to get the symmetry breaking across the connectivity. Remember how I was talking about symmetry breaking generates manifolds. We know this is important. We know it is a determinant. So from my own personal perspective, these are the two elements I would like to see being under control, because it is those that determine the dynamics we see in the brain imaging. Yeah? And this is our board of evaluations, because if the disease does not express itself in the brain imaging data are useless anyway. Yeah? I would like to be able to exploit to a maximum what is identifiable in the brain imaging.
Thank you, Victor. I think we're going to break now. I think Victor at least needs coffee. So you're a little bit behind us actually on common. Let's start back at 10:45, so about 25 minutes. Thank you. The coffee up there for people who are here actually, people who are there virtually, too bad. See you at 10:45.